This is Chelsea Holman with an overview of my first lab activity from the perspective of a student. The lab activity that I chose to do was from Online Chem Labs. It was specifically the DNA hydrolysis lab. The lab can be found at www.onlinechemlabs.com. You will be required to set up an account, and from there you can choose your labs. I do highly recommend that you complete the lab techniques lab first. This lab gave an overview of how all the labs are set up with intro material, then the lab activity itself with follow-up questions at the end. It also introduces you to the lab interface and the different tools that are going to be used and how those tools are used electronically as opposed to in real life. Now, the goals for this activity are threefold. The first is to identify nucleobases by thin layer chromatography. The second is to learn how the DNA is broken down into individual bases by hydrolysis. And the third is to identify which bases are most easily hydrolyzed. Now, the four bases, just for a quick review, are A for adenine, G for guanine, T for thymine, and C for cytosine. Now I will take you through the lab activity itself. There are 3 parts to the lab, as I said before. The introduction, which contains all of the background material that you will need to know for the lab itself. The second is the experiment, where you will be performing the actual tasks and recording data. And the third asks you some questions as well as draw conclusions. The introductory material covers the following. It starts with explaining what DNA is and how it is organized. DNA is a double-stranded helix, which is then wrapped around histones. Each individual strand is made up of nucleotides, which are held together by phosphodiester bonds. These phosphodiester bonds can be broken through hydrolysis, which is one point of the lab. The nucleotides themselves are made up of a base bound to a ribose sugar ring, which is phosphorylated. This bond between the base and the ribose sugar can also be broken through hydrolysis. This is an image of the lab setup, or the so-called remote labs environment. It contains the following. In the upper left-hand corner is the control dispensers. This has 3 control bases for reference. Underneath that are the TLC plates. Next to this is the plate reader that gives the output on the monitor. The next row contains the trough, which contains the solvent for the thin layer chromatography. There are also capillary tubes for transferring samples. There is a pipette for delivering, in this case, hydrochloric acid. Next to that, in the microcentrifuge tube, is the DNA sample itself, next to a centrifuge, which is not used. Then at the bottom is the oil bath, which is set for 95 degrees Celsius, along with a timer that is used for timing all of the incubations in the oil bath. Finally, there is a trash can. In this lab, thin layer chromatography is used to separate out bases from a mixture. Now, for thin layer chromatography to work, a plate or TLC plate is dotted along the starting point line with the different mixtures. It is then placed inside of the trough where the solvent will rise up the plate by capillary action. The top, when it reaches the top of the plate, that is called the solvent front. Now, once it is read, the distance that the solute, or the parts of the mixture that we're interested in, have traveled gives us information. So the most important piece of information that you get is the RF value. And the RF is defined by the distance that the solute has traveled over the distance that the solvent front has traveled. This is demonstrated in this image where the starting point to the solvent front is demonstrated in green, and the starting point to the solute distance is demonstrated in red. This gives us an RF value. Now, this RF value for controls can be compared to samples, and that will give us information about what solutes are present in our sample mixture. Now, this is an example of a plate image from the actual experiment. We were given G, A, and T controls to compare our sample to. You can see the solvent front is indicated by the red arrow. The dots on the far left is where the sample was originally dotted, and the dots throughout the distance in between are where our solutes have landed at the end of the experiment. Now, we can easily glance at this and notice that the sample seems to have traveled the same distance as A, which leads us to believe that our sample contains free adenine base. However, it is always best to calculate the RF values for each sample as well as each control. Once the RF values are determined for this solvent, then the controls do not have to be rerun, even though the instructions do emphasize that you should rerun them. Next, we were asked to incubate the DNA sample at 95 degrees Celsius for 5 minutes and run another thin layer chromatography plate. There was no change, and once again, only A was identified. Next, 1 ml of hydrochloric acid was added to the DNA sample and then again incubated for 5 minutes at 95 degrees Celsius. And again, a third thin layer chromatography was run. This time, you can see that there are 4 dots in the sample. This indicates that all 4 bases are present in the sample. After this experiment was completed, a series of questions that require critical thinking were asked. These included, what is the 4th spot that doesn't correspond to any of the controls? The correct answer is the base cytosine, because C was not given to us as a control. Next was, what would you conclude if after 12 hours of more heating, no dots were observed? The correct answer would be that the compounds had broken down even further to small molecules that could not be observed by thin layer chromatography. What would you see if all glycoside bonds in the DNA were hydrolyzed? The correct answer is you would see bright, high concentration dots for all 4 bases. Which base is most readily hydrolyzed? This would be adenine A, which was the dot that we saw in the very first example. Now, a few notes about the lab. The lab could be repeated, although while you could repeat the experiment, the answers in the experimental section could not be changed. However, the conclusion section questions could be changed up until the time that the instructor closed the activity. The full activity took me about 30 minutes, although for someone that was not familiar with the background material as well as the techniques involved, it could take up to an hour, I would say, but no more. There weren't really any wet lab techniques that were required. Because everything is automated, the actual way that you go about using the equipment is somewhat different. I have to say, I had a lot of issues with the way that the pipettes out 1 ml, but there's no transferring solutions from one location to another, and the pipettes are just preloaded with the solution that you need. This actually made me rethink my opinion that distance labs can fully replace classical in-person wet labs. There is really a lack of good lab technique being taught because everything is being done in a computer interface, which is understandable, of course. There was not really enough material covered, in my opinion, to write a full lab report. Like I said, it only took me 30 minutes. There were only 3 samples that were run. I felt like there were a lot of other variables that could have been explored and a lot more depth that the lab could have gone into. The first thing that they tell you at the beginning of the lab is that they're not going to show you the DNA extraction because it would be too difficult to incorporate into a computer interface type lab. It would be interesting to have a quick video or demonstration of some kind, even if you don't have to be performing the task yourself. There just needs to be something to better explain how that extraction happens rather than one quick paragraph in the introductory material. Now, I did run into one technical problem. The timer and the oil bath were supposed to be linked so that the tube of the DNA sample could only be removed once the eject button popped up. The eject button was only supposed to pop up after the full 5-minute incubation. However, what I observed was after only 90 seconds, the eject button pops. And if you click on that, it didn't get its full 5-minute incubation, and the computer knew that it didn't get the full 5-minute incubation, yet allowed you to remove the sample. Then once I put the sample back in, by just adding 2 different times in the water bath, which added up to a total 5 minutes, was not enough. A full 5-minute incubation without interruption needed to be obtained in order to get the correct results. This was just it wasn't that big of a problem, but I have to say that the directions specifically tell you to try to remove the sample ahead of time, and notice that it won't allow you to. But because of the technical problem, you were able to, and therefore that screwed up your experimental design. Just some final thoughts that I had about the experiment as a whole. I didn't really feel that it covered enough material. There were, like I said before, a lot of other directions that the experiment could have gone in, other variables that it could have covered. It also could have gone into more details in the concept, specifically in the introduction. It spent a lot of time talking about DNA, but very little time talking about the actual chemical reaction of hydrolysis, the way that acid will affect hydrolysis reactions. I also thought the directions were very clear. However, it left no room for exploration in terms of why don't we allow the students to have this interface and add acid, heat for different amounts of time, see what happens, get fresh DNA samples, and do things in different orders. If you heat and then add acid and don't reheat, what happens? It was very clear cut in terms of what you were supposed to complete to answer the questions, and didn't really veer outside of that at all in terms of teaching students the scientific method or the practices that we had talked about before. Overall, I would say that it was a good lab. It got the point across. However, I don't know that it could truly replace a classical lab face-to-face environment.